I'm just going through the component 2 paper with you that we have just done in class. Should be in front of you and marked. Now, those of you who've got the A star, please feel free to just watch selected bits or just work with the mark scheme where you see fit and as you see fit, because whatever you're doing is obviously working. Those of you that are struggling a little bit, please go through this with me. I'm going to go through question by question, flicking over to the mark scheme all along so that you know what wording we're going to need to hit. It's a jumping the hoops exercise. You need to know where your hoops are. Right, first one. It's a question about insects and their larvae, and larval stages do look a bit similar. So how do you identify the larvae and put them into the individual species? How do you classify them into the individual species instead of just, okay, it's small and it's in a stream? And the keyword that you've got here is protein analysis. Quite a few of you went into the direction of DNA and genetics. It asked you specifically protein analysis. How do you know by what proteins you've got in which larval stage, which species it could be in? If you know the protein, you will know what amino acid sequence it will be. Proteins are made out of amino acids. If you know what amino acid sequences you'll find in which species, some proteins may be unique to that particular species, or you may just have that and that protein in this species, but no others. So linking that to known proteins that are known to be in this particular species, that's what I've got you the 2 marks here. So compare the amino acid sequence. Protein analysis means look at what amino acids do you have there against known sequences, so sequences that you know are identified in specific insects. That would have gotten you the 2 marks for that one there. The second question, it starts to literally give you that whole scenario. So it is giving you the background of you in a mountain region in South America, a lovely graph on the higher you go in that mountain range, the lower your temperature of the water in the stream will be. So again, if you get a graph, read the labels. It gives you some background information. And again, it gives you even more background information about different streams at different heights, similar pH, similar rocks, but it's still giving you an idea as to what investigation's actually been done. It is describing kick sampling to you. I know that we've had kick sampling in a previous question somewhere, might come again. Now, the question doesn't actually refer back to the scenario. That's kind of keep that in the back of your mind. The question here is, why do you kick sample upstream and upstream and upstream and upstream? Why don't you kick sample the same place? Why don't you kick sample downstream? Why do you kick sample upstream? That kind of goes into thinking, right, you're kicking loose a rock or a few larger pebbles to see what little species float out and float downstream in the water. Oh, gravity is pulling the water downstream on that mountain. So the species will be pulled downstream and your net will be placed further downstream. So if you then have a second sample, if you do that further downstream, you might still have species that were kicked loose, floating and being caught in the net for your second kick sample. So this is linking together that gravity makes the water flow downstream and everything that is already kicked loose flows downstream, therefore will affect your second sample if it's taken further downstream. Hence, do it upstream. Mark scheme is wording it that way around. Uncaught organisms would flow downstream, and therefore working upstream would prevent more organisms being caught than actually would be in that second kick sample. You can rephrase that in various different ways, or back to front, front to back. That's okay. I can still give you the marks for that. The next question here, suggest why the quadrats were placed in the middle of each stream. Why were the quadrats or the nets placed in the middle of each stream? The wording here is kind of making you think, hold on a second, what are you trying to avoid? You're trying to avoid what is happening on the edges of the stream, on the banks, on the edges of the stream. So what have you got in the middle of the stream? Perhaps you've got a stable temperature, stable water flow rate, but whatever you're not wanting is whatever the effect of the edges is. Maybe you've got soil input from the side, maybe you've got fluctuating water, maybe you've got shading by trees by the edges of the stream. So whatever the effect of the edge is, you want to avoid it. The mark scheme is wording it, minimize the effects of the banks, similar flow of water, similar light, and I can accept quite a range of ideas there, as long as you've got the idea that we're avoiding the edge of that the stream. The following question here gets you into the Simpson's Biodiversity Index. So it's giving you 3 different heights, and it's asking you to calculate out the one at height 3,000 meters. So it's got those numbers here. It doesn't ask you to calculate all of them it gives you numbers later on. But the highest one up in the mountains is giving you here. How many for each species were caught. It actually, even on the next page, beautifully gives you the capital N definition and lower N definition for the total number of families. That's your total number of families, okay? And then N-1, so whatever that number was, N-1 from there, times N with N-1, and you should be getting for that to your 462. To get to your sum of lowercase n times n-1, that was your individual. How many do you have? N-1, and then n times n-1. And yes, if you're timesing by 0, it's going to be a 0. The adding that column here up, I can't select by itself, it seems. Adding that column on the edge up is going to give you the total that you require here for your lowercase n times n-1 all summed up, and that should give you the 124. From there, the question is actually quite beautiful. It gives you the equation. You're lucky that year it gave the equation. Sometimes it doesn't, so be prepared for it to not be as nice as this. 1 minus your sum above, divided by underneath, and you should have gotten from that your 0.73. If you're stuck, just give me a shout. I'll come over and I'll look at what you've done and see where the little mistakes do creep in. Okay, so don't beat yourselves up. I can very often, even if you've calculated your numbers on the previous page wrongly, I can give you the correct follow-on marks. Even if you carried over a mistake, you carried it over correctly, it gives you the marks. The next question here, it gives you already calculated numbers for the biodiversity index. Here it gives you the 0.7 and 0.8.